In this video, I will tell you which computer vision approach is best for suiting different pathology image analysis problems. When do we use semantic segmentation? When shall we just use object detection? And when do we need instance segmentation? Hi, I'm Alexandra Zhurev, and I'm here to help you do better digital pathology. This video is a part of a webinar I co-hosted with an image analysis company, Aiforia. Aiforia uses artificial intelligence for image analysis, and we were talking about different tips and tricks for annotations for developing deep learning models. I'm going to leave the link to the full webinar in the description below, but now let's get into matching the correct computer vision approach to the different pathology problems we are trying to solve with image analysis. Last thing that we're going to talk about is that no one size fits all for pathology problems. Different pathology problems require different image analysis solutions and annotation approaches. And what do I mean by different image analysis solutions? I am referring to computer vision way of solving problems. So we had this example where we wanted to localize and delineate as well as quantify the area of those tumors. And we annotated in a certain way. And Lindsay mentioned this is semantic segmentation. Semantic segmentation is the way computer approaches this problem. But if we're doing this, we are not counting. This is just to detect the regions and to quantify the area. If we want to count, then we are using so-called object detection. And if we are doing semantic segmentation, we're not doing object detection. If we're doing object detection, we're not doing semantic segmentation. So you need to think about what your pathology problem is and then use the correct way of solving it. It ties to the setup of your project in Aiforia or whichever software you're using, but basically the concept is the important thing. But obviously, there are problems in pathology where we want to have both, right? What do we do when we want to have both? When we want to have, like, let's say, each red blood cells delineated or some other cell delineated. We want to count them and we want to know the area and we want to know everything. There is a solution for that as well. Like in this case, we want to count this cell. We have this object detector that we already know from the previous project, but we want to also delineate it. We want to know the shape, we want to know the area, maybe there is some hypertrophy, some other processes, shrinkage, whatever. We want to know the area and we want to have both. And this is called instance segmentation. And there is an option to do instance segmentation to have it all in Aiforia. And you would ask, why can't we like always have it all? Basically, it's easy for us to see. It's not so easy for computer to see. So each of these tasks requires some computational resources. And if you don't need to use all the resources, then just don't use them because it's going to slow down your project. It's going to make it more expensive. So basically, the previous knowledge, which computer vision approach to use for your pathology problem is going to guide the setup of your project and is going to make your analysis more efficient. And if you want to learn more about that, I'm going to send you to another resource. I'm going to send you to my blog and to my YouTube channel, where we have a video about what instance segmentation is in tissue image analysis. And we have all of the examples of other ways of detection of semantic segmentation with a video showing this on a different project in Aiforia platform as well. This was actually the last tip that we gave in the webinar. So if you want to know all the tips and tricks, be sure to click the link in the description below and register for the on-demand recording of the webinar. I'm looking forward to seeing you there.